Welcome back. We will solve 3 very important questions from CSR Net question papers. We'll solve them one by one. So let's get started. The first question says, the transport of fructose into the enterocytes is mediated by option 1, sodium dependent glucose transporter 1, that is SGLT1. Option 2, glucose transporter 5, that is GLUT5. Option 3 is GLT2 and option 4 GLUT4. So among these 4 we will have to choose the correct answer. Now quickly recall what we have learnt about transporters. Then we will be able to solve this question very easily. Monosaccharides are taken up into the enterocytes. Now, glucose and galactose are taken up by sodium glucose cotransporter 1, that is sodium dependent glucose transporter 1 or SGLT1, that is active carrier transport is seen here. It transports sodium along with glucose or galactose. And fructose is taken up by facilitated diffusion through GLUT5. Now, glucose, fructose and galactose are transported out of the enterocytes into the capillary. That is when capillary is absorbing it. It happens through GLUT2. So, our correct option is option number 2, glucose transporter 5 or GLUT5. Now coming to the next question, the di and tripeptides are transported in the enterocytes by peptide transporter 1 that requires option 1, sodium, option 2, calcium, option 3, hydrogen and option 4, chloride ions. So among these 4 you'll have to choose the correct answer. As we all know, the apical membrane of the small intestinal enterocytes possess an uptake system for di and tripeptides. They enter the enterocyte cytoplasm through peptide transporter 1 or T1 as stated in the question. Now it requires sodium hydrogen or hydrogen sodium ion exchanger. Now single amino acid move from the lumen into the enterocyte by facilitated diffusion in sodium linked transporter. But for di and tripeptide transport, hydrogen ion symporter is needed. Now this di and tripeptide and hydrogen ion enter into the cell in the same direction. That is why we are calling it as symporter. Remember, secondary active transport occurred through sodium symporter and active transport of amino acid occurs through amino acid pump. This is primary active transport. Amino acid pump means what? Where ATP is needed. It is primary active transport and this happens in the apical surface. And amino acid is absorbed through the basal surface by facilitated diffusion, which is a passive process. And in the basal surface, sodium pump is needed, which is again an active process, that is active transport. Coming to the correct answer, so our correct answer is option 3, hydrogen ion. Going to the next question, insulin increases facilitated diffusion of glucose in muscle cells by 4 options are given. You'll have to choose the correct option. Option 1, phosphorylation of glucose transporters. Option 2, translocation of glucose transporter containing endosomes into the cell membrane. Option 3, inhibition of the synthesis of mRNA for glucose transporters. And option 4, dephosphorylation of glucose transporters. GLUT4 is the insulin responsive glucose transporter that is responsible for postprandial glucose clearance. In muscle and adipose cells, GLUT4 is packaged in vesicles that are released upon stimulation of the insulin receptor, and in case of muscles, it also happens when you are exercising, that is in response to exercise. The mechanism for GLUT4 is a very good example of cascade effect, where binding of ligand to the membrane receptor amplifies the signal and causes cellular response. Here what we get to see is trafficking of GLUT4 containing vesicles towards a cell periphery, docking at exocytic sites and plasma membrane fusion. So from here we can see that what is happening is clearly stated in option 2. Translocation of glucose transporter containing endosomes into the cell membrane. Cell membrane means here plasma membrane. So what is happening? When there is binding of ligand, then what is happening? The vesicles where these transporters are packaged, they are translocated to the plasma membrane and they fuse with the membrane. That means GLUT4 is translocated to the cell membrane. The glucose transporter is translocated to the cell membrane or plasma membrane, and this happens after binding of a ligand to the membrane receptor. So our correct answer is option 2.